Right. So now here that when you're talking about embryology, we have to talk about gonads. So gonads are primary sex organs, right? So primary sex organs. So these are we call ovaries or testes. So ovaries presented in females and testes present in males. So male gametes. So male gametes we call sperm, right? Female gametes we call ova or eggs, right? A gamete is the female or male reproductive cell that contain half the genetic material of the organism are produced through the process of meiosis. So there are two type of cell division, meiosis and mitosis. Meiosis means reduction division. So in our testes and ovaries, so only meiosis occur. All other cells in the human body, when they are living, so they undergo mitotic cell division, right? So now here, this is the normal representation of the human development, normal development of the human, right? So zygote, so this is fertilization, then you get zygote, then four-cell, then you get morula, blastocyst, embryo. So after that, embryo grow to fetus and eventually you get a baby, right? So now here, developmental stage start from cleavage until ninth week, right? The embryo first undergo division without growth. The embryo enters the uterus at 16 cell stage. So you need to remember embryo. So that means after fertilization. So we fertilize at the top of the fallopian tube. So one third from the one third, that means towards the ovary. So it is fertilization occur in one fallopian tube normally. So then uh, zygote forms there, right? So then embryo enter the uterus at the 16 cell state. So uterus, some people pronounce as uterus, but uterus, right? Fetus. Right? In human development, fetus is prenatal human between its embryonic stage and its birth. The period from the beginning of the ninth week to birth is known as fetal period, right? Not a fatal period, fetal period. It is characterized by maturation of tissue and organs and rapid growth of the body, right? So look at this one now here. This is the embryo, this is fetus. So normally embryonic period, so it's nine, that means about 8 weeks, right? So 3 to 8 weeks. So this is fatal period. So it is here 9, I'll tell why it is, right? So there's another term you have to know about that, conceptus, right? So include all the structures that develop from the zygote. So these terms you all have to know what is zygote, right? Both embryonic and extra embryonic. So that means it is in that is embryonic, you divide it into embryonic and extra. Extra embryonic means so maybe as associate. So uh, associate structures that you see from the outside, right? Denote the embryo, its adnexa, appendages or adjunct parts or associate membrane, placenta and yolk sac. So those things you go to breed, right? Okay, next part, stages of prenatal development, right? Stages of prenatal development. So it is A, B, C we have categorized as A, B, C. So A we call germinal period, right? So first 2 weeks after conception. Embryonic period, 2 to 8 weeks after conception. So C, fatal period, 2 months to 7 months after conception. So you need to remember this one now here, germinal, 0 to 2 weeks. That means so you just fertilize. Then after that, embryonic, 3 to 8 weeks, and fetal. So 9 weeks birth. So, right. So now you have to know about what is gestational age. Gestational age, right? Gestational age. Gestational age is the common term used during pregnancy to describe how far along the pregnancy is. It is measured in weeks from the first day of the woman's last menstrual cycle to the current date. A normal pregnancy can range from 38 to 42 weeks. Infants born before 37 weeks are considered premature. Right. So then you have to know about this one, trimesters or trimesters, right? Pregnancy is measured in trimesters. Oh, some people pronounce as trimester, it doesn't matter. So it's simple, first trimester, right? So week 1 through 12 weeks, that means first trimester, week 1 to 3 months, right? Week 1 to 3 months. Oh, right, week 1 to 3, that means up to 3 months is better. Second week, week 13 to week 27. So what does it mean? 4 to 6 months, right? Third trimester, so 7 to 9, that means just close to the birth, no? Third trimester pregnancy span from week 8 to the birth, right? Trimester. So that these are important in, recording, gin and obs recording. So you write is first trimester, second trimester, right? But these are not very accurate as you see. It depends on the mother's condition. These are average values, right? So next one is the abortion. This is the broad term, abortion. Abortion is the ending of pregnancy by removing a fetus or embryo before it can survive outside the uterus. An abortion which occurs spontaneously known as miscarriage. An abortion may be caused purposely and then called induced abortion, or less frequently induced miscarriage, right? So abortion is the main term, right? So abortion, so in Sri Lanka, abortion without doctor's consent, that means if it is harmful for mothers and babies' health, abortion is prohibited by Sri Lankan law. But in certain judiciaries, in other countries, it is allowed up to a certain age level of the developing baby, right? So abortion means ending. So that means you can divide into two. One thing is a spontaneous abortion. That means miscarriage. This term we have heard. Other one is the you purposely end this one. So we call induced miscarriage or induced abortion, right? So there are so many sub varieties about this abortion, right? So many names. Right, abortion is a really controversial topic. It has been discussed and it is debated topic. So especially induced miscarriage or induced abortions. Right, now let's watch a video to understand about little bit more information about abortion.
Abortion is the termination of pregnancy. Embryo. The rate of miscarriage risk is high at the start of pregnancy. Mostly, miscarriages happen in the first trimester, that is, the first 12 weeks of gestation period. The earliest weeks of pregnancy, before the 20th week, is the time when a woman is at the highest risk of a miscarriage. The risk starts to drop when pregnancy reaches the 6th week. It is better to keep in mind that complications can arise at any point in the whole pregnancy period. Classification of abortion. Early abortion. It is the removal of undesired tissues in the first trimester in the 10th to 14th week of confirmed pregnancy through pills. Late abortion. The abortion after the 24th week of pregnancy is called late abortion. Spontaneous abortion. The death of the fetus before 20 weeks of pregnancy is spontaneous abortion. Causes of spontaneous abortion. It can happen due to multiple reasons. There are no specifically mentioned causes. A spontaneous abortion does not mean it was caused by something you did. A list of problems that result in spontaneous abortion given by daily medicos are several viruses such as cytomegalovirus, herpes virus, parvovirus, and rubella virus. Chromosomal and genetic abnormalities. For chromosomal and genetic abnormalities, there comes intrauterine fetal demise, stillbirth, blighted ovum, no embryo formation, molar pregnancy, no fetal development, partial molar pregnancy. Defects in phases also cause miscarriages. Immunologic abnormalities, major trauma, uterine abnormalities like fibroids, adhesions also cause spontaneous abortion, placental defects, retroverted uterus, cervix problems, hormonal problems in mother, infections, uncontrolled diabetes, thyroid disorders, abnormal fetal growth. These errors in genes and chromosomes of the mother or father can occur naturally and promote no fetal growth. Damaged cells, embryo division, or damaged sperm cells are also natural causes of abortion. Induced abortion. The intentional removal of embryo in case of unwanted pregnancy is induced abortion. Threatened abortion. Threatened abortion is marked by vaginal bleeding that occurs in the first trimester, which could be a sign of possible miscarriage. Inevitable abortion. The symptoms of inevitable abortion are intrauterine pregnancy with worse cramps, high vaginal bleeding, and cervix dilation that results in miscarriage. Complete abortion. Complete abortion happens during the 8th week when all the tissues leave the body and pass through the vagina with bleeding. Incomplete abortion. When some of the tissues leave the body and some products stay in the body, this is incomplete abortion. Recurrent abortion. 3 consecutive miscarriages in 20 weeks is considered as recurrent pregnancy. Missed abortion. When the fetus didn't form and has died but the tissues remain in the uterus is missed abortion. Septic abortion. A uterine infection can spread to the placenta and fetus before and after induced abortion and can develop sepsis in the uterus, placenta, fetus, and vital organs. Risk factors involved in abortion. Risk factors of spontaneous abortion can be avoided to prevent abortion or miscarriage. Certain risk factors that could lead a woman to miscarriage are: risk factor of abortion increases with age greater than 35. History of spontaneous abortion. Tobacco consumption, use of alcohol, drugs, that is cocaine. Chronic disorder in the mother, that is diabetes, hypertension, overt thyroid disorders. High caffeine intake, uterine and cervix problems, excess weight, invasive prenatal tests, poor diet or malnutrition, advanced maternal age, abnormally shaped uterus, severe high blood pressure, food poisoning, exposure to toxins like arsenic, lead, etc., use of certain medications. Prevention of miscarriage. Some tips for avoiding a miscarriage are: regular exercise like jogging, walking, or cycling. Regular prenatal follow-ups. Avoid smoking, drug use, and alcohol intake. Intake of prescribed multivitamin. Limit caffeine intake during the pregnancy, but it's better to avoid it. Maintenance of a healthy weight. A healthy and balanced diet with all nutrients and minerals in every meal. Avoid things that might develop infections. Add fruits and vegetables in your daily diet. Maintain a happy and calming environment. Find ways of managing your stress. Diagnosis, confirmation of abortion. The tests that are carried out for diagnosis and confirmation of abortion are transvaginal ultrasonography, transabdominal ultrasonography, serum HCG tests. Treatment of abortions. Commonly for women who have an abortion except complete have 3 treatment managing options. Expectant management. Doctors wait for the rest of the tissue to pass out naturally in this type of treatment. Medical management. This treatment option requires medicines to remove the remaining tissue out of the body. Surgical management. This treatment option requires a surgical procedure to remove the tissues remaining, if any.
Thank you for watching our video.
Right. So now continue from here. So now you realize, so it has so many varieties. That means different types, right? Coming back to the session, right? So then you can, when you're free, you can study more about abortion to get more idea, right? Next one, right? So next one is gametogenesis. Gametogenesis, right? Gametogenesis is defined as a process of formation, right? Process of formation of gametes, right? It's a normal thing, right? In gonads, gonads means that we discuss what is gonads, right? So next one, right? Derived from primordial germ cells during fourth week of IUL. IUL means intrauterine life, right? So spermatogenesis, formation of male gametes, right? So formation of the male gamete, spermatozoa, sperm in the male gonad testis. Oogenesis, formation of female gamete, secondary oocyte in the female gonad ovary. So now I think that in A-level classes also you might have encountered these things. So in your A-level courses, even in O-level also, you might have given some basic knowledge of this one. This is spermatogenesis, that the process in which that you make sperms. Other one is the oogenesis, so that eventually you get the oover, right? You can see now here when you look at these two diagrams, there are significant difference between spermatogenesis and oogenesis. Significant difference than spermatogenesis and oogenesis, right? Okay, now here, what we are going to talk about, spermatogenesis. Spermatogenesis, right? So spermatogenesis, so this is also process, right? So that we learn about meiosis, right? Mitosis. So these two type of main cell division, mitosis and meiosis. So mitosis means that you inhale. So from here to here, you get mitosis division. Mitosis division means the daughter cells also get the same genetic composition. Meiosis means you get chesmata crossing over, so you get reduction. That means if it is, 4 chromosomes here, you get 2 each, right? So meiosis is important for genetic variation, right? So lower animals and plant normally, they follow uh, asexual reproduction via mitosis, right? Conversion of the spermatogonia. Spermatogonia is 2N, right? So to spermatozoa, sperm is N. Spermatogonia remain dominant in a seminiferous tubule till puberty. So at 11 or 10, 11, 12 only the spermatogenesis begin, puberty, and continues till old age. So there are reports that even at 80 years or 90 years, some healthy males can generate fertile sperms, right? So some more information, spermatogenesis. So spermatogenesis occur in two phases. Phase 1, cell growth and multiplication from primordial germ cell till spermatid formation. Phase 2, spermatogenesis, cell metamorphosis, process by which spermatids changes into spermatozoa, right? So time of puberty, testis canalize to become seminiferous tubules, right? So you need to understand what is canalize, right? I'll explain that in later, right? Primordial germ cells give rise to spermatogonia sperm cells. These cells formed at regular intervals, Ka type A spermatogonia. So you can research and find about now spermatogonia. So from the spermatogonia A, so it could go for the clone cell, and also it go to the spermatogonia B. These cells formed at regular intervals, Ka type A spermatogonia. This begin the process of spermatogenesis. They undergo meiotic division and the last produce type B spermatogonia. So you can research and see more about the Ka type A spermatogonia and type B spermatogonia, right? So type B spermatogonia then enlarge and get transformed into primary spermatocyte, diploid chromosome. So this is the diploid cell for 44XY, that means a 45, 46, right? Each primary spermatocyte undergo first meiotic division. So we call reduction division. So you get 22X. So you know X and Y chromosome. So that 50% sperms, you get X chromosome, and this is the sex chromosome, and another 50% you get Y chromosome. So when the X, but it's an ovum, it is definitely X. So when X sperm and the ovum fuse, so you get a girl, and the Y sperm joined with the ovum, that you get a uh, uh, male, right? Each second spermatozoa haploid undergoes second meiotic division from two spermatids. Totally, so you get four spermatids, right? Two with the X and two with Y chromosomes are formed. The entire process spermatogenesis, so it called 2 months to complete. So this is the spermatogenesis process. So that means if you get two primary spermatocytes, so you get what? So, 8. So this is meiosis 1, meiosis 2, you get a 8, because why? Two cells. But from one cell, you get, right, from one cell, you get 4 spermatozoa, right? One cell. So two cells, so it is 8. So you have to know that type B spermatogonium and type A spermatogonium. So I explained little bit, because type A spermatogonium, so can be a clone cell, and part of it again go for type B spermatogonium through intermediate spermatogonium, right? Next one, spermatogenesis. So you can see spermatogonium type A. So it go that means another direction for what does it mean. So it is for the cloning, right? So another path, but type A become type B, right? So now here the cells belong to one type A. See, spermatogonia form a clone. They are all connected to each other. So that's what I said, making clone. So cells are embedded in the Sertoli cell. So Sertoli cells give nourishment. So this is in A-level syllabus function of the Sertoli cell. Sertoli support, protection, nutrition, and discharge of mature spermatozoa. Right. Spermatogenesis is managed by two hormones, right? One is LH, luteinizing hormone. So stimulate the ladies to secrete testosterone, which promotes spermatogenesis in Sertoli cells. FSH, folic stimulating hormone, also influence the Sertoli cells. So these two major hormones. So that means if you have, some problem in this hormone secretion, so you may delay or you may not get spermatogenesis in males. The process of transformation of spermatids to spermatozoa called spermiogenesis. So this term is embryological term. So normally, some of you might not have heard about spermiogenesis. Spermiogenesis. So that it's like a metamorphosis. You might have heard about how the butterfly, the metamorphosis of the how the step forming a butterfly. So it just like there are so many events happening. One thing is condensation of the nucleus. So this is the nucleus. So this is the initial, right? Initial stage. So then what happened? Formation of the acrosome. So this is now here the acrosome cap developing here. Then what happened? Formation of the neck, middle piece and tail, then shedding of excess cytoplasm, because you don't want more cytoplasm. So here in the middle piece, if you look at this one, in the middle piece, you get a lot of mitochondria, because why you need mitochondria? So that sperm has long way to go for fertilization, so it gives you energy, right? About 300 million spermatozoa produced daily. That is a huge number, right? Maybe I think larger than population of America, right? So sperms are not motile when stored in epididymis, but become motile after ejaculate only. So that epididymis is this, no? Epididymis, right? And the number of motile active sperm per ejaculate also influence the fertility. So normally sperm count vary 20 to 50 million per milliliter. Right, so the semen enough for normal, but normally if it is more than 20 million per milliliter, it is better. But if you get sperm count less than 10 million, you sterilize. That means under normal condition, uh, you can't give a baby. Means sperm is not enough to penetrate the egg cell and make a zygote. But still, you don't have to worry about that, because why now there are so many methods that even you can get the one good sperm. And so there are methods now you can artificially fertilize and get babies. But those are again against the nature, but it is happening at the moment, right? So structure of mature spermatozoa, right? So head, you get nucleus, actosome cap also there. This is a little bit more advanced than A level. Actosome cap is there. So it's better you need to, because everything we can't say here that because no time. So you need to study more detail, watch more videos. So I'll give you the list of reference, right? So then a neck has proximal centriole. So it's single we call kendra there, from which egg cell filament starts. Middle fish mitochondria encircle egg cell filament. So it's a middle fish. So here tail. Proximal principal piece. Distal end piece. So this is the typical that's drawing of the matured sperm, right? But if you study about, because there are science that there are embryologists who extensively study about the sperms, right? There are so many morphological variation even in the healthy sperm. So you can specialize if you like embryology, so you can study about this thing, right? Okay, so this part is the what we discussed today is the right. What is it? Spermatogenesis, right? So spermatogenesis. So in the next lesson, we are going to talk about, right, oogenesis, right? So what I want you all to do today, right? So I'll share the video. So please make about 2-page note about spermatogenesis, right? So in next day lesson, we are going to talk about oogenesis, right?